What's your current employment?
Okay, so I'm actually a post-PhD in a glycomev laboratory, an academic laboratory associated to one university, and previously I performed a PhD about endocrine secretion. So first, I would like to thank the Spim organizing committee for giving me the opportunity to present our results concerning the effect of urban air pollution on the physiology of human skin epidermis. So this FUE project is the product of the collaboration of two academic laboratories, Glycomev Laboratory and URCOM, and 4 private companies, Uriage, BioEC, BioEurope, and Genex. Air pollutants can be divided in 2 classes. A little part of primary pollutant is due to natural events like volcanoes eruption. But most of primary pollutants are due to human activity, like, for example, road traffic and industrial activities. The secondary pollutants are the results of chemical reaction between the primaries. A lot of studies were focused on the effect of pollutant on lung physiology, but our epidermis, as the first line of defense of our organism, is also continuously exposed to pollution. So in this study, our aim was to better understand the effect of urban air pollution on skin physiology. If we consider the number of abstracts correlated with pollutant and skin in PubMed database, we observe that only 183 abstracts are available. So our knowledge about pollutants' effect on skins remains limited. Most of previous studies were focused on the effect of only one independent pollutant. So, I'm sorry, I think there is a problem. Okay, so in the study, our aim was to mimic pollutant exposure as close as possible to the reality. So the first challenge was to develop a model which mimics this reality. So BioEC developed an innovative tool named Polybux, which allows us to expose human skin explants to a mixture of environmental pollutants. In this manner, only the stratum corneum is exposed to pollutants. The composition of the mixture was defined using AirParif database, and this mixture is composed of volatile organic compounds, polycyclic aromatic hydrocarbons, heavy metals, and particulate matter. Skin explants are maintained in culture during 5 days. The treatment, the pollutant exposure is performed during 90 minutes, and then analyses are performed 1 or 2 days after the exposure. In this talk, I will present you a transcriptomic approach and then a microscopic analysis. So first, I will begin with the transcriptomic analysis. Genex laboratory performed a microarray analysis of whole skin explants, and they used total RNA to this study. They showed a significant modulation of expression of detoxifying enzyme genes and the activation of AHR and NRF2 pathways after pollutant exposure. So we decided to focus our attention on 13 genes for the continuation of the study. This scheme presents the gene we decided to study. So hydrocarbons activate the aryl hydrocarbon receptor, which after activation is translocated in the nucleus to bind DNA xenobiotic response element domain and induce the transcription of genes encoding for proteins involved in aromatic compounds degradation, like cytochrome P450. This degradation enhances reactive oxygen species production, which activate NRF2. NRF2 will be phosphorylated by several kinases and translocated in the nucleus to induce the transcription of genes encoding for proteins involved in ROS elimination and toxic compounds elimination. The red circles represent the gene we decided to study. So I will present you the results we obtained after laser capture microdissection. As I said previously, GenX identified 13 genes we decided to focus on, we decided to study. So in our lab, we are able to isolate a basal and granular keratinocyte using laser capture microdissection. And we studied the modulation of gene expression using RT-qPCR. This picture represents the technique. The region of interest is cut with a laser and recovered in a tube cap. After qPCR analysis, we didn't show any significant modulation of gene expression in stratum basal, whereas we showed a significant overexpression of CYP1B1, HMOX1, UGT1A6 and UGT1A8 in stratum granulosum enriched fraction. So the pollutant exposure is able to induce a significant overexpression of CYP1B1, HMOX, UGT1A6 and UGT1A8 in stratum granulosum enriched fractions. To confirm this data, we decided to study at the protein level, the effect of pollutant exposure at the protein level, and we performed some immunolabeling experiment. First, let me present you the protocol to prepare a skin explant for electron microscopy. So skin explants were exposed to pollutant mix or to organic solvent. Then they were cryo-prepared after embedding, and ultra-thin section allows us to perform immunogold to detect the AHR localization. Image analysis allows us to determine the proportion of AHR in the nucleus versus the cytosol of 10 cells per epidermal layer. This is a typical picture we can obtain after electron microscopic observation. The white circles represent the gold beads, which represent AHR, and we quantify the number of beads in the nucleus and in the cytosol. In this manner, we were able to quantify the translocation of AHR in each epidermal layer. We won't show any significant differences in stratum basal and stratum spinosum, whereas we showed a significant translocation of AHR in stratum granulosum. So hydrocarbons pollutants are able to activate cytosolic AHR and induce its translocation into the nucleus. To further investigate this analysis, we performed immunostaining of protein identified in microarrays. Here you can see that MAFG, a transcription factor associated to NRF2, and AKR1C3, an aldoketoreductase, are overexpressed 2 days after pollutant treatment. qPCR results were also confirmed at the protein level using immunostaining. Here you have the staining of CYP1B1, involved in oxidation of several compounds, including polycyclic aromatic hydrocarbons. And you can see here that the labeling is higher 2 days after the pollutant treatment. HMOX1 is also overexpressed, but 24 hours after treatment. Finally, UGT1AA, which is involved in conjugation and elimination of toxic xenobiotic and endogenous compounds, is overexpressed 1 and 2 days after pollutant treatment. Then we decided to focus our attention on diesel particle penetration. First, we isolate corneocytes using tape stripping approach, and we observed using electron microscopy that particles smaller than 300 nanometers were able to penetrate in corneocytes. Then we observed ultra-thin section of skin explants, and we observed diesel particles up to the granular layer. So particles smaller than 300 nanometers penetrate through the corneocytes and granular keratinocytes in our model. Since Genex's laboratory put in light an alteration of the barrier function of skin after pollutant exposure, we decided to focus our attention on lipid distribution in cell. So I will present you preliminary results we obtained concerning this lipid distribution. Using Niloid dye, we observe the lipid distribution in the epidermis, and as you can see here, when the label is high, the color is green, and when the label is low, the color is dark or dark blue. And after pollutant exposure, we observe a lipid reorganization, an alteration of lipid distribution. Using line scan analysis, we were able to show that this reorganization was significant at the stratum granulosum. To better understand this process, we observed lamellar bodies because we thought that this lipid reorganization could be due to an alteration of lamellar body secretion. After electron microscopy observations and image reconstruction, we showed no significant differences in the number of secreted lamellar bodies at the interface of stratum granulosum and corneum. So, modification of lipid distribution does not seem to be related to alteration of lamellar body secretion. Other investigations are in progress to better understand this pollutant effect on skin lipid distribution. To conclude this talk, we can say that in our model, pollutant exposure modifies the expression of genes encoding for detoxifying enzyme like CYP1B1, HMOX1, UGT1A6, and UGT1A8 in stratum granulosum. Pollutant exposure induces an activation of IHR and its translocation in the nucleus in the stratum granulosum. Pollutant exposure also makes diesel particles to reach the stratum granulosum, and pollutant exposure alters the epidermal lipid distribution at the junction of stratum corneum and stratum granulosum. To end this talk, I would like to thank all our partners, especially Yasmina, who performed all electron microscopic observation, Marine, who performed qPCR analysis, and Marie-Laure, who managed this project in the lab. Of course, I thank our sponsors, and I thank you for your attention. If you want to further discuss about pollutant exposure and skin physiology, you can meet us at the poster station. Thank you.
Thank you very much. Do you have any questions? Yes, please. Over there. On the other side.
Thank you very much. Very interesting. So if I understood you correct, your selection of genes was based on primary hypothesis driven. So you had like differently expressed genes, and then you took one that all followed the same pathway, right?
Yes.
And do you have any plans with the other differently expressed genes to work out how they contribute?
We have to select some genes because the technique of laser capture microdissection doesn't provide a lot of samples. So we use microarray approach to screen the response at the transcriptomic level and focus our attention on a few genes due to technique limitations.
But you still may have a lot of interesting genes in your data set.
Yes.
Thank you. Other questions, please? Yes.
Regarding your lipid data, did you use, do you know the dye that was used? Is it a BODP dye to label the lipids in the stratum corneum? We
will perform several experiment, and BODP is one of the dye we wanted to use to confirm the results we obtained with nitride.
Because, so just as a comment, it could also be that the pollution or the exposure modifies the lipids in a way that they don't bind anymore to the dye, so that could somehow hamper the analysis.
Yes. Well, I present you preliminary results. It's just the beginning of the story.
Well, just following on this question, have you seen any differences between the kinds of pollutants that you've used on the distribution of lipids? Because I can guess that some solvents, for example, would introduce different changes than particles from diesel, for example.
Yes. Our model is based on the exposure with a mix of pollutants. So we can't study pollutant individually, but we compare pollutant exposure to solvent exposure, and the pictures I've shown was the result of this comparison.
Okay. Well, electron microscopy was not done by you yourself.
Yasmina and me performed the...
Yasmina and you. Because, well, there is a kind of question I would like to ask about the diesel particles. Mm-hmm. It seemed to me that they were inside the cells of the stratum corneum. Is it right?
Can you repeat?
The particles, I mean the carbon particles,
yes,
seemed to me on your picture to be inside the cells, yes, in the stratum corneum.
Yes, it's right. We
How do you explain this? Because, as you may know, keratinocytes in the stratum corneum are just cornified bags, which have no activity of membrane as living cells. How do these particles get in?
It's a very good question. I don't have any idea. For the moment, I can't answer this response. I'm sorry, but the only things we have shown is these particles in corneocytes. But,
well, I can understand that. Yeah.
But it's interesting to note that in the granular layer, we observe that particles seems to be in a kind of vesicle, but I don't know how.
Well, it depends on the time of exposure, of
Yes,
course,
maybe.
because you can imagine that those particles would migrate into the stratum granulosum, then be absorbed by the keratinocytes,
Exactly.
and then desquame, but it would take 2 weeks.
Yes, but in the experiment using tape stripping, we use pollutant mixed enriched in diesel particles, so it's very artificial method to see the particles. We are in the kind of real exposure in the picture with skin explants.
Okay. Yes, please.
In the back. Actually, I had the same question, but I was wondering, the particles you see by EM are found in like free in the cytosol, or kind of encapsulated in vesicles, or because they are rather large. 300 nanometer is rather large. So
Yes, if I. Here you can see the particles in the corneocytes, and we can't see any vesicles. So, but here in the skin explant section, we can see a kind of membrane around the vesicles, and it's also what it's shown in literature. So I don't know if I answer your question.
Just thank you for your talk. So, do you know the medium size of your particles?
I don't know the medium size, but I know that particles smaller than 300 nanometers were able to penetrate the corneocytes.
Do you think that it could cross against the membrane through any kind of transporter of channel or any kind like that that could explain and could reconciliate everyone around the table for the discussion about the absorption of your particles?
Yes, of course. I don't know the size of molecules which can...
Or specific transporter or something like that. I think that you could check that, because so you probably get so much this kind of question in the future.
Okay, thank you.
Well, thank you very much for your talk. And I call the next speaker, who will be Magdalena Plotczyk, the role of human hair follicles in scar modeling. Yes, please. So, Magdalena Plotczyk comes from the United Kingdom.
Yes. So I'm a third-year PhD candidate in the lab of Dr. Claire Higgins in the Department of Bioengineering at Imperial College London. So I would like to thank the organizing committee for giving me the opportunity to present today. Yes. So today I'm going to tell you about my research looking at the role of human hair follicles in scar remodeling. So let me first introduce you to hair follicles. And hair follicles are these unique mini organs that undergo constant cycles of growth. And because of that, a single hair follicle can produce many fibers throughout the years. In addition, the surrounding skin is also changing. And the changes include remodeling of collagen and increasing vascularization and thickness of epidermal tissue when the hair follicle is growing. So interestingly, in the case of injury, it's the same mechanisms that are involved in wound healing and are responsible for skin repair. However, in most cases, wound healing results in the formation of a scar. And despite differences in different scar types, all of them are characterized by an abnormal deposition of large amounts of collagen and lack of functional structures such as hair follicles. So now, would it be possible to reverse this process of scar formation by reintroducing a growing hair follicle in order to promote the remodeling changes that we can see in a hair follicle cycle in healthy skin? So in order to answer this question, we've designed a clinical study in a collaboration with a hair transplant surgeon, and we transplanted hair follicles into stretch scars. So these scars are formed in 5% of hair transplantation surgeries when a strip of skin is excised from the back of the head and in order to harvest follicles to be transplanted to the front. So these stretch scars are a great model for our study because they are all formed in the same place, all on the back, on the head. They all are formed in males only and all because of the same surgical procedure. So in our study, we collected a 3mm biopsy from a scar before transplantation, and then at 2, 4, and 6 months after the transplantation. So we know that a growing hair follicle can induce remodeling changes in the healthy skin, but we don't know whether it can also affect scar tissue. And also, despite many studies being published on scar tissue, most of them are focusing on pathological scarring, such as keloid or hypertrophic scars. So because stretch scars have never been studied before, I've decided to characterize them and compare to the healthy neighboring skin. So we know that there are differences in the composition of extracellular matrix and the network in scarring. So I decided to look at collagen type 1 fibers, and I used second harmonic generation imaging to do that. And in this process, two photons interact with a unique medium, such as collagen type 1, and these fibers have a lack of central symmetry. And thanks to that, photons can interact and combine and create a one second harmonic generation photon that is twice the original frequency and so twice the energy. So this imaging system is great because there is no need for staining, and we can image quite deep into the tissue without compromising resolution. So I can then detect this signal using high sensitivity detectors, and I can scan through quite big areas of the tissue. So after image acquisition, I used a simple 2-channel segmentation to separate the fibers from the background. But I found that it didn't really represent the architecture of collagen. So for that reason, I used an alternative technique, a tool called Elastic. And I trained the software to recognize 3 different features. So there were thick collagen fibers above 10 microns, thin fibers below 10 microns in the background. And thanks to that, I separated all my images into 3 channels, which represented the architecture much better. So using batch processing on 45 images, I analyzed collagen fibers in both conditions, and I found a significant increase in the collagen density in stretch scars, which was represented by an increase in the total percentage of collagen and lower fiber spacing. In terms of the thickness, there were significantly more thick collagen fibers in stretch scars, while the average thickness of fibers in both categories was significantly higher. I've also looked at the orientation, and I created this color-coded maps of orientation angles, and I plotted the distribution of these angles. So what we can see here is that in scars, the fibers peak at 0 degrees, meaning they all run more or less parallel to the epidermis, while in healthy skin, the angles are much more spread out. So I also quantified the alignment coefficient, which tells us more about the alignment of fibers in relation to each other. So because the previous image analysis can be highly affected by fiber density, I've decided to use another method. And this is based on the intensity of the second harmonic generation signal. And this paper shows really nicely that when all the molecules in the tissue are nicely aligned, this gives us the maximum second harmonic generation signal, while partial alignment gives us much lower values. So I quantified this in my tissue, And I found the intensity of the second harmonic generation signal to be significantly higher in stretch scars, meaning the fibers were more aligned. So I was also able to detect the autofluorescence signal coming from elastin fibers using the same multiphoton system. And I found that the proportion of elastin as compared to collagen type 1 fibers was significantly lower in stretch scars. So now, given that there are differences in the extracellular matrix, I suspected that it might have an effect on the mechanical properties of my tissue. So I used atomic force microscopy, and in this test, we indent a cantilever towards a section of the skin, and we measure the deflection of this cantilever, which tells us more about the stiffness of our tissue. So I compared the stiffness in healthy and stretch scar tissue, and I indeed observed areas of much higher stiffness in the scars. So I also looked at the cellular content of my tissue, and I found that epidermal thickness and dermal cell density were significantly lower in stretch scars. But we know that dermis is very heterogeneous. So in the next step, I'm going to look at the differences in the cell type composition of the two conditions. I was also interested in the level of vascularization, which I found to be significantly lower in stretch scars with no difference in the vessel circumference. And what's interesting here is that this is in opposite to hypertrophic and keloid scars, which can actually show significantly higher vessel density than the healthy skin. So when unfortunately we cannot see this, but you can come to my poster. So when we look at the epidermal-dermal junction, this characteristic reed ridges that we can see in epidermis will not longer present in scars. Okay, so we know now that there are big differences, structural differences between stretch scars and healthy skin, but I wanted to know how they affect the gene expression. So I collected RNA from the dermis of stretch scars and the dermis of healthy skin, and I've performed an unbiased transcriptomic analysis using a whole genome microarray. So I found 200 genes that were differently and significantly expressed between stretch scars and healthy skin. And we can see an area of 106 genes that were down and 94 genes that were upregulated in stretch scars. So let's look at the cellular distribution of the protein products of these 200 differentially expressed genes. And we can see that there are some transcriptional regulators, there are some cytoplasmic proteins and plasma membrane proteins. But because we know that there are major differences in the extracellular matrix of the stretch scars, I was particularly interested in the proteins deposited outside of the cell. So through literature research, I found 3 proteins that were particularly relevant in wound healing and scarring, and we've heard about them today. So it's thrombospondin 1, thrombospondin 2, and ADAM metallopeptidase with thrombospondin motifs. So I first validated this finding using immunofluorescence staining for thrombospondin 1, and I found a similar trend with downregulation of thrombospondin 1 in stretch scars. And I next isolated fibroblasts from this excised tissue, and I isolated scar fibroblasts, papillary fibroblasts from the top 400 microns of the healthy dermis, and reticular fibroblasts from the lower part of the healthy dermis. So I first tested the expression of my selected targets, and I found a significantly higher expression of thrombospondin 2 in papillary fibroblasts as compared to scar fibroblasts, while the expression of ADAM TS12 was significantly higher in scar fibroblasts as compared to both the healthy fibroblast types. So this data suggests that the phenotype that I observed in vivo is actually partially preserved in fibroblasts that I isolated and cultured in vitro. So in my future experiments, I'm gonna try to look for the extent of this gene expression differences and test the expression of other genes that are regulated downstream by thrombospondins. So I also looked at the behavioral differences of these fibroblast types, and I found that the population doubling time of scar fibroblasts was significantly higher as compared to other fibroblasts, which means that this fibroblast proliferated the slowest. And this can possibly explain the in vivo phenotype of reduced dermal cellularity that I observed in stretch scars. And in order to test the effect on promoting vascularization of different fibroblast types, I collected a conditioned media from them, and I applied the conditioned media to endothelial cells. And I found that there was that scar fibroblasts proliferated significantly lower endothelial cell proliferation than papillary fibroblasts. So once again, this might suggest the link between my in vivo phenotype of reduced reduced density in stretch scars. Okay, so to summarize this part of my talk, so far I showed that there are major structural and gene expression differences between stretch scars and healthy skin, and some of these differences are actually preserved in fibroblasts isolated from the tissue. So this allows me to go back to my original question, which is whether hair follicles can induce remodeling in scars. So I analyzed samples from before transplantation and after transplantation, and I looked at 3 patients with stretch scars. And what's important here is that all the scars were established, meaning they were 4 years old or more, and we assumed that at this point the tissue reached homeostasis. And they were also of a very similar width. So I applied the same collagen analysis pipeline, and I saw no difference in the total percentage of collagen and only a small drop of fiber spacing at 2 months after transplantation. However, when I look at the thickness, there was a significant increase in the proportion of thin collagen fibers after transplantation, while the average thickness of fibers in both categories was also reduced. So this reminds us about a shift towards a healthy collagen phenotype. What I also found was that the fibers became more disorganized with a higher spread of orientation angles at 6 months, which was also supported by a drop in the collagen, in the alignment coefficient. And the remodeling of collagen was also visible looking at the intensity of second harmonic generation signal, which was significantly decreased at 4 and 6 months after hair follicle transplantation, once again showing a shift towards a healthy phenotype. I've also looked at the cellular content, and I look at the cellular content at a minimum distance of 200 microns away from the transplanted follicle. So I wanted to make sure that I'm testing only the host tissue. So I found that the epidermal thickness and dermal cell density significantly increased at 2 months, and they actually stayed upregulated at 6 months, which once again reminds us about the phenotype of the healthy skin. There was also a decrease in vascularization at 4 months with no difference in the vessel circumference. Again, healthy phenotype. So I've also observed a gradual change in the initially flat epidermal-dermal junction with restoration of reed ridges that are characteristic of the healthy skin. Okay, so now we know that when we transplant a hair follicle into a stretch scar, there are major structural changes towards a healthy phenotype, but I wanted to know more about the mechanism by which it happens. So I collected RNA from the dermis of my samples, and I performed another unbiased transcriptomic analysis using a whole genome microarray. And I found over 1,700 genes that were differentially expressed after hair follicle transplantation. And we can see on the heat map, we can see an area of genes that were primarily upregulated and downregulated after the transplant. So having a closer look into these genes, we can see that there were 266 genes that were upregulated after transplantation, and even after 6 months after transplantation, they were still upregulated, and Similarly, 294 genes got downregulated and stayed downregulated. Looking at the cellular distribution of the protein products of these differentially expressed genes, we can see a high level of interaction between them. So in my future experiments, I'm planning to look into these interactions in order to understand the molecular mechanism by which the remodeling happens. But another question that remains now is whether this hair follicle-induced remodeling is unique for stretch scars. So I tested that in a case study of a patient with a 50-year-old scar on his thigh, and this scar was completely devoid of hair follicles, and it also resulted from his thigh being a source of a skin graft for another side of the body. So I applied. So first I compared the initial characteristics of stretch scars and skin graft scar, and I found that the epidermal thickness and vessel density were significantly higher in stretch scars. And that could be explained by the proximity of hair follicles surrounding the scar. So we know that scars can be highly heterogeneous and the baseline levels can be very different. But would this also would it also influence the effect of transplanting hair follicles? And to answer that, I've applied the same collagen analysis pipeline, and I found a similar remodeling shift towards a healthy phenotype with a decrease in collagen thickness and higher fiber spacing. So looking at the cellular content, there was also an increase in the epidermal thickness and dermal cell density, as well as vessel density after hair follicle transplantation, once again reminding us about the healthy skin phenotype. So to summarize, in my talk, in my research, I wanted to know whether transplanting of a hair follicle into stretch scars can remodel the tissue and improve its phenotype. But because my model system, stretch scars, haven't been studied before, I decided to first characterize them. And I showed you significant structure and gene expression differences between stretch scars and healthy skin, with some of these differences being preserved in fibroblasts isolated from the tissue. So next, I looked at samples from a scar before transplantation and then after transplantation, and I found that some of the structural differences between scar and healthy skin are actually changing towards a healthy phenotype. So in my future experiments, I'm planning to combine the structural observations and the transcriptional analysis in order to identify the biological processes and the underlying molecular mechanism by which a hair follicle can induce remodeling in scars. And hopefully, in the long term, the results of this study will allow me to identify the target, which we can use to design therapeutic strategies to reduce scarring. So I would like to thank my supervisor, Dr. Claire Higgins, who's here in the middle, my whole lab, the imaging facility at Imperial, our clinical collaborators, and my sources of funding. Thank you.
Thank you very much, Magdalena. Do you have questions to the speaker, to the presenter? Well, think about this, and I'll ask the first question. The density of hair follicles that you graft is always the same.
Yes, it was also always the same, and it was reflecting the same density that would be used for hair transplantation surgeries.
So the influence that you suspect for the modification and reversal of scarring, it does come from the grafted cells, probably.
Yes, definitely. So hopefully by investigating the different targets that I found in the microarray, I'll be able to translate this into the cell-based experiments and in a year I would tell you exactly which cells and how it would be contributing to the observed effect.
Thank you. Dr. Marley has a question for you.
So, very nice presentation, thank you, very stimulating. I was wondering if you could consider that air grafting, I don't really know how it's performed, but can air grafting be considered as a wound itself, a new wound that you create in the previous wound somehow, and can it contribute to trigger a remodeling response in this way, or is it just...
Yes, so that's a great question. So there is a procedure called microneedling, which is claimed to improve scar phenotype or appearance of the scars. However, unfortunately, there is not much literature concerning the molecular effect of microneedling. But if I can refer to some of the previous studies. So in this study, a clinical study compared the effect of hairy punch grafts with non-hairy punch grafts, and the author showed that the hairy punch grafts promoted wound healing better than the non-hairy punch grafts. So we believe that like the hair follicles are actually the active ingredients that can change the skin. And that's something I'm gonna check using fibroblasts isolated both from the skin and from the hair follicle in order to test how potent they are in remodeling and changing the skin.
Yep. There are no... Yes, Lionel? Last question, please.
Thank you for your talk, and congratulations. So, just to go back to the genes that you screen across this method, so did you find some special thing around repressive genes that could be controlled by this kind of approach? I'm thinking, for example, of myelin or something like that. Do you check this type of gene?
Could you repeat the question?
Myelin. M-E-A-N-D-E-N. Myelin. You know?
Myelin?
Yeah.
Oh, myelin for myelination.
Myelin, like the repressive gene in wounds described by people from the Gallo Labs and the Scripps Institute in San Diego, for example.
I mean, it's an interesting suggestion, but I haven't come across any genes like that in my lists. But I'm definitely looking closer into the different possible mechanisms, which is almost a never-ending list of possibilities.
Yes, because it could be an option in the future to create.
Yeah, so definitely, I'm looking for suggestions. So thank you for this.
Thanks a lot. Thank you, Magdalena.
Thank you.
The next speaker would be Joya. It comes from Lyon. LBTI, which is part of IBCP Institute, CNRS, and University of Lyon. She works with Dr. Debray.
Yeah, yes.
And she's a PhD student.
Okay, so good evening. First of all, I would like to thank the scientific committee for giving me the opportunity to present you my work today. So in this work, we established a new role of GPRC5A in the keratinocyte mechanotransduction. Before to start, I would like to introduce some key notion, which is about epidermis homeostasis. So the homeostasis epidermis, the epidermis homeostasis depend on several characteristics. This including the balance between the proliferation and the differentiation, as well as the well organization of the epidermal cells through the intercellular junction. Moreover, the epidermis homeostasis is closely linked to the mechanical properties of its underlying tissue, which is the dermis. it has been described that the the dermis mechanical properties is around 5 to 10 kilopascal. So it has been also described that the extracellular matrix stiffness affects many functions, processes. So in skin, the stiffness promotes the proliferation, the migration, and also the wound healing processes. But whereas the stiffness inhibits the differentiation and the adherent junctions formation, and at the end alter the skin barriers. Concerning the wound healing, it has been shown that the stiffness is strongly associated with a better skin regeneration. In fact, Goffin showed that the wound granulation tissue is increased during the wound healing from 18 to 30 kilopascal. Moreover, injection of the hydrogel with different stiffness in the wounded skin in mouse show that an optimal stiffness is necessary to improve the wound healing process by promoting fibroblast transformation, wound healing, wound closure, and the keratinocyte proliferation and migration. Moreover, many studies have shown that the keratinocytes are able to sense and respond to the mechanical changes. In our laboratory, we also demonstrated that the human keratinocyte also responds to the stiffness substrate. To do that, we use the polyacrylamide hydrogel, in which the concentration of the acrylamide and the bisacrylamides are changed in order to obtain different rigidities. And in order to promote the keratinocyte adhesion, the gel surface was functionalized with the type 1 collagen. Using 3 different rigidities that we know that we call soft, medium, and rigid, compared to the glass cover slip, we show that on the soft hydrogel, the keratinocyte cell shape remain round and no clear focal adhesion points was observed. This morphology is associated with an inhibition of keratinocyte proliferation. When we observe keratinocyte morphology on the medium, the rigid, and the glass, we observe that the cells were more spread with a well-defined focal adhesion point. This morphology is associated with a keratinocyte proliferation. Moreover, we also demonstrated that the cells in the soft didn't proliferate, but when these cells were replaced on the plastic surface, a favorable condition to proliferate, the keratinocytes are able to proliferate again. In the same way, we also measure keratinocyte colony formation efficiency. We observe that during the 2 second generation, the keratinocyte from a soft hydrogel decrease the colony formation compared to the keratinocyte from glass. This observation was reversed during the 3rd generation. So our question is by which mechanism the keratinocytes are able to re-engage their proliferation. So our objective is to identify the actor that involved by the keratinocyte mechanosensing in response to the stiffness. So we perform RNA sequencing where we compare the profile expression of the keratinocyte culture on the glass cover slip, on the hydrogel with a soft rigidities, and the condition where the cells were replaced that we named the soft glass condition. This ambient approach reveals a strong signature of the glass condition compared to the soft and the softer glass. When we analyze the gene profile expression, we observe many genes are differentially expressed. Despite a strong signature of the glass condition, when we compare the softer glass versus glass condition, we still find 43 overexpressed and 62 repressed genes. Focus on making of these genes potential candidates to in keratinocyte mechanosensing. Focus on the overexpressed gene, we identify a potential candidate, which is GPRC5A, for G protein coupled receptor class C, family 5, member A. We choose GPRC5A because of its high expression during the transition, and because GPRC5A is a receptor, and we know that the mechanotransduction starts at the plasma membrane. We next confirm that GPRC5 expression is correlated to the stiffness changes. So to do that, we culture the keratinocyte on the different hydrogel, and we replate them on the glass condition. We observe that GPRC5A expression was always overexpressed during the transition from the hydrogel, so the soft, medium, rigid, to the glass surface. To rule out the GPRC5A expression due to the replating process, we include the glass-to-glass, and in which no significant expression was observed. We can note that the more the difference between initial and final substrate, the stronger the expression of GPRC5A. We also demonstrated with this result that GPRC5A expression is correlated to the stiffness changes. To illustrate these stiffness changes, we focus on the wound healing context, where we know that we observe an increase of the stiffness. We analyze the expression of GPRC5A in the wounded skin of mouse model. The immunostaining revealed that a specific expression of GPRC5A at the leading edge, the place of keratinocyte migration. We also observe that when we look in the region far from the margin and in homeostasis condition, GPRC5A is not detectable. This observation led us to study the role of GPRC5A in different cellular processes, such as the adhesion and the migration process, which is the main process that occurs during the repetitilization phase. We analyzed GPRC5A relocalization during the early phase of adhesion, proliferation, and the differentiation. Our study shows a diffuse expression of GPRC5A when the cells is in suspension, an extreme soft condition. When the cells were plated and adhere in the surface, we observe a part of GPRC5A is translocated to the nucleus during the first phase of adhesion and during the proliferation. At the differentiation state, GPRC5A expression in the nucleus returned to the cytoplasm. Making of this result, regarding of the result, we also would like to, identify in which compartment GPRC5A is expressed. Regarding to the nucleus, we show that GPRC5A co-localizes with the nucleolin, which is a specific nucleolar protein. Concerning the cytoplasm expression of GPRC5A, our immunostaining revealed that GPRC5A co-localizes with the endoplasmic reticulum with the vesicular trafficking, but not in the Golgi apparatus. This shows that GPRC5A translocation in the nucleus is correlated to the early adhesion process. To explore further the adhesion process, we repress GPRC5A expression using the siRNA approach. First of all, we verify that GPRC5A expression is repressed by qPCR and Western blotting. The analysis of the adhesion assay revealed that the assay GPRC5A transfected cells show a better adhesion compared to the control. We also analyzed keratinocyte migration, and we show that the assay GPRC5A transfected cells delay the closure compared to the control. This result proves the causality between GPRC5 expression and the adhesion and migration process. We also analyzed keratinocyte differentiation process. A limitation of the siRNA approach is the fact that they are transient. To get rid of this problem and to study GPRC5A involvement and the process and the long process, such as the differentiation, we established a stable cell line of immortalized keratinocyte entered, in which the expression of GPRC5A is silenced by shRNA. Of note, we have validated all of the previous results regarding to the adhesion and the migration process on the entered cell line. Our analysis of keratinocyte differentiation process in the 2D models show that the GPRC5A inhibition exacerbates the expression of marker differentiation such as the keratin 10, the involucrin, and loecrine. To go further in keratinocyte differentiation, we took advantage of 3D model of reconstructed epidermis. First of all, we analyzed GPRC5A expression during epidermis formation. The immunostaining revealed that GPRC5A is expressed during the first week and after is decreased during the stratification. This transient expression of GPRC5A suppose a role of this protein during the first step of keratinocyte differentiation. To test this hypothesis, we perform a reconstructed epidermis with SH-GPRC5A, and we analyze epidermis morphology at day 6. As you can see, with the hematoxylin and eosin staining, we show that in the SH-GPRC5A condition, we can observe two different areas. The first areas, we show that the thickness of the epidermis is decreased compared to the control. And in the second areas, we observe a disorganized epidermis with a detachment of the suprabasal layers. When we analyze a junction and the junction protein and keratinocyte differentiation markers protein, this immunostaining confirmed that the differentiation process is impaired by strongly reduced of eucadarin, as you can see here in green, and keratin 10, and also a discontinued expression of the involucrin in this condition. Moreover, the tunnel assay revealed a high proportion of apoptotic cells in the disorganized epidermis. To conclude, we demonstrated that in homeostasis, GPRC5A is not expressed. However, a disturbance of this homeostasis during the wound healing, in which the dermis mechanical properties is increased, this increase induces expression of GPRC5A, which may in turn increase the capacity of keratinocytes to migrate, reduce the cell adhesion in order to restore a functional epidermis. We also demonstrated that GPRC5A inhibition in the stiff condition show an alteration of keratinocyte differentiation, process, and this could lead to a disorganized epidermis. We can suppose that when the dermis mechanical properties is abnormal, like a soft substrate, the GPRC5A expression is reduced, and this can contribute to a disorganized phenotype. Finally, we can also hypothesize that in the soft substrate, where GPRC5A is reduced, a GPRC5A rescue could lead to restore the epidermis functionality. In light of this result, we think that GPRC5A may be a key mechanotransducer, helping the keratinocyte to adapt to mechanical changes in different physio and pathological contexts. Finally, I would like to thank my supervisor, Romain Debré, Bérangère Fromy, and Philippe Faure, and also to thank the FDTC team, and thank you for your attention.
Thank you, Chua. Do you have questions for Chua? Yes, over there, please.
I have two questions. This protein, GPRC5, is a GPCR, isn't it? Is a GPCR?
Yes, it's a GPCR.
So this is a transmembrane protein?
Yeah.
So I was just curious how these kind of proteins can reach the nucleolus. And the second question would be, do you know if there is any relationship between forming this focal adhesion and the activity of this GPCR, or vice versa? If they have something to do together, or it's totally unrelated?
Alors, concerning the first question, the localization of the, in fact, GPRC5A is a G protein coupled receptor. And we know that this type of receptor is located at the plasma membrane. But another studies reveal that show GPRC5A localization into the cytoplasm also. Concerning the nucleolus localization, we used two different antibodies to visualize GPRC5A. One target the N-terminus and the other target the C-terminus. And we show you here is an antibody against the C-terminal. And we think we have a cleavage of this part. And is a short C-terminal of GPRC5A translocate in the nucleus. And the second, can you repeat the second question, please?
No, you're right, it was not totally clear. Because when the cells are on a stiff substrate, they start to make this focal adhesion, and these focal adhesion are mechanotransducer. So I was just wondering whether activity of a GPCR signaling whatever, and the force that can be transmitted to the cytoskeleton, if there is any relationship. For instance, if you, knock down your GPCR, do you prevent the formation of focal adhesion or vice versa?
We don't see the focal adhesion after the inhibition. We just analyze their capacity to form a the early adhesion. So I don't know if I answer your question. Yes.
Next question, please. Anybody would like to comment or ask something? Well, do you consider the bottom of the wound, any wound, to be a stiff substrate? I mean, you suggest that the keratinocytes would colonize the wound bottom.
Yes.
The bottom of the wound would vary according to the clinics, whether you just remove epidermis, whether it's farther deeper in the dermis. So probably the stiffness of this substrate for keratinocyte colonization would also change. How do you position your model towards this clinical reality? I mean, how to put this different way? You suppose that the keratinocytes would spread on the rigid surface, is it?
Yes.
But this rigidity varies according to the clinical situation. I mean, sometimes it's only just epidermis that is removed, and you have still basement membrane, for example. When the wounding is deeper, the mechanical properties of the wound, of the bottom of the wound, would change. So do you have any idea how the modification of the wound, epithelial wound healing would be affected by those mechanical differences between different wounds? No, not yet.
I think no, but if you want, we can discuss after, because I.
One-to-one.
Yeah.
Face to face, you know.
Just face to face.
Well, okay. That was the first step of your study, and then you can develop on this. Okay. Any questions?
Yes.
Yes, Lion.
Only one. So, thank you for your talk. So, great hypothesis. So, just to know if you plan to check something in differentially display between chronic wounds and acute wounds, to know if this kind of process is present or not in chronic disease without no.
I'm sorry, can you just speak a little bit higher?
Okay,
Yeah.
I will do. I understand why you mentioned to speak face to face. So, I just wondering if you plan to check in the lab of yourself the difference between expression or these global fields in chronic versus acute, because in some chronic wound disorders, there is nothing that could be control the wound healing process. And do you think that this kind of target could be involved or not in this way?
It's for us. It's difficult to check this expression in another wound healing.
Chronic, long-lasting.
Yes, because we don't have the sample.
But
you should get it, and you can do.
Leg ulcers are the chronic wounds.
Yeah.
So it could be one of the possibilities to study.
Okay.
Could be. But I let you engage talk with. Sorry, but so it's really nice. Concrete.
Okay. No more questions? Okay, no more answers. Thank you very much.